the disease. Yep, in progress. And genetic screening. So X-linked Dystonia Parkinsonism presents complex challenges that touch the lives of patients, families, caregivers, and healthcare professionals. As we embark on this educational journey, we aim to illuminate the various facets of this condition, from its intricate genetic underpinnings to the latest advancement in diagnosis and treatment. The partnership between the PNAA and the Collaborative Center for X-linked Dystonia Parkinsonism, or CCXDP, signifies a shared commitment to enhancing awareness, knowledge, and patient care. Through the webinar series, we endeavor to bridge the gap between scientific understanding and practical application, providing a platform for fruitful discussions, valuable insights, and exchanging ideas. We are honored tonight to have distinguished panel of experts, clinicians, researchers, and you can see them, and advocates who will guide us through the complexities of X-linked Dystonia Parkinsonism. So before we start the education part, we will be hearing an opening remark from our lovely president from PNAA, Dr. Gloria Briones.
So good evening, everyone, and I welcome you to the first series of this CCXDP called Dystonia Parkinsonism, which is endemic in Aklan, western region of the Philippines, Visayas region, and where I am, being an Ilongga. This is really dear to me as a Filipino nurse, that we can, the PNAA, can raise awareness to all of us about this disease. I remembered when I was a little girl seeing a couple of people around me with dystonia, Parkinsonism. Then I didn't understand. So tonight, we are privileged. This collaborative partnership with the Collaborative Center for CCXDP, which is the General Massachusetts Hospital in Boston, and PNAA have this partnership. So without further ado, I just want to welcome each and every one of you, and please continue. There are 2 more series to come in September, and there is another one, but there are 3 series. So tonight, let's learn more about this disease condition, Dystonia Parkinsonism. Thank you.
Thank you, Dr. Briones. So before we start, I just want to remind everybody just some few housekeeping. So first, make sure you mute your phone so that, we can hear loudly or clearly our speakers for tonight. Second, use the chat. So if you have feel free to use the feature to interact with fellow attendees, say hi, probably include where you're coming from, what time zone you are in, and ask questions throughout the webinar in the chat box. Although, third, we have a Q&A session that will be facilitated by our president-elect, Dr. Marlon Saria. So make sure that, you put that question in writing and ready to ask that away after the session. And then engage on social media. So our lovely webinar, the Zoom master, Carol Robles, is always making sure that, because we're focusing on awareness about X-linked Dystonia Parkinsonism. So make sure you shout it to the world. Let's make sure that everybody's aware of this condition. Be respectful in the environment and stay for the duration. Hopefully we'll learn a lot, continue to discuss about this. 7, recording and materials will be we are recording, so it will be available. So technical support, if you're encountering any issues, just reach out to our Zoom master. and participation and networking. We expect you to ask questions and be engaged to tonight's session. And last but not the least, feedback. So at the end of the session or the webinar, you will receive a survey. And please take a few minutes to provide your insights so we can continue improving for this webinar series. So without further ado, this is the time. I will let each of our panel of speakers and experts to introduce themselves because, if I will introduce them, 10 minutes is not enough for each speaker. That's how amazing they are. So Chris Bragg, Dr. Chris Bragg, you may start.
Hi, everyone. I'm Chris Bragg, and I'm here with my colleagues Nutan Sharma and Ozilius, who will introduce themselves. And we are from the Collaborative Center for XLinked Dystonia Parkinsonism, based in Boston. And we're very happy to share our work with you tonight. Nutan, do you want to say hi?
Yes. Good evening, everyone. So I'm a movement disorder neurologist specializing in relatively rare genetic forms of dystonia and Parkinson's. And I have worked with Chris and Lori for over 20 years in this area. Go ahead, Lori.
Hi, my name is Lori Ozilius. I'm very pleased to be here. I'm a geneticist, also at Mass General, and I have my main focus is different forms of dystonia, but I dabble a bit in Parkinson's and essential tremor.
Okay, and how about I share my screen? And that's okay. Okay. And all right. So again, thank you, Mary Joyce and Dr. Baronis, for your kind introductions and the partnership. CCXDP is really honored to be working with the PNAA. We truly believe that your organization can partner with us in our mission to help spread the word about what X-linked Dystonia Parkinsonism, or XDP, is, which can help families who may be at risk recognize it and connect them with the resources that we can provide. I'm going to say just a few words about us, and I'm going to turn it over to Nutan. We were founded January of 2014, so we are almost 10 years old, when a group of individuals came to us and told us about this disease that affects uniquely people from the Western Visayas region of the Philippines, that they care about tremendously, and they want us to go out and find a cure. And so with their partnership over the past 10 years, we've gone about building a very substantial global network of scientists and clinicians and patient families and patient advocates, all working together to try and find a cure for this disease. We have strong partnerships in the Philippines with most of the major medical centers based in Manila. We have created infrastructure in Panay, where most known patients currently reside, that we provide comprehensive free medical care to patients with XDP and support services to their families. And what's shown on this slide are all the institutions around the world that make up our research portfolio. We give a lot of money away in grants to get people who've never thought about XDP to bring us their ideas and help us find a cure. And it's truly been a very exciting journey for us in this. We've learned a lot about this disease. We've learned about the genetic cause, and you'll hear about that tonight. We've learned a lot about the symptoms and have some ideas about how to treat it. But our goal is to find a cure, to find effective therapies that really help people regain their lives. And so you're going to hear about where we are on that journey tonight. So I think with that, I'm going to turn over to Nutan, who's going to talk about the clinical features.
Okay. All right. So what is X-linked dystonia Parkinsonism? Over the years, multiple names have been given to it. So just the abbreviation XDP. It's also referred to as DYT3 because there is a nomenclature for identifying genes that cause dystonia, and this was number 3 in the list. And then locally in the region in the Philippines, where many of these men live, it has been referred to as LUBAG over the years. So this is an adult onset disorder. We think onset begins in the early 30s. So these are males who develop normally, and then they develop, and then in adulthood, they develop features of dystonia and Parkinsonism. As far as we know, all of the men have maternal ancestry that can be traced to the island of Panay. And the first cases were identified by Dr. Viterbo, an internist in Rojas City, and he reported these cases in the literature in 1976. And on the upper right, you see the drawings depicting these men, because this was prior to the age of, videos, mobile video cameras, and it was just drawings and photos. And so what are the clinical features? First, there is dystonia. Dystonia is defined as the contraction of muscles that are both agonist and antagonist muscles that contract at the same time, and they result in abnormal postures. You can have dystonia that only affects one body region, and it's called focal, or it's called segmental if 2 or more body regions are involved. It is multifocal if there are 2 or more body regions involved that are separated, let's say a right foot and a left hand. And generalized dystonia means by definition that at least the trunk and 2 other body regions are affected. So dystonia describes the clinical feature, the uncontrolled contraction of muscle that leads to the abnormal postures that you see in these drawings. So the trunk can be flexed, the foot may be bent, or the big toe may be sticking up. The hand and arm may be bent uncontrollably, extending behind the body. Commonly in XDP, we see that the jaw opens and there's abnormal, uncontrolled twisting of the neck. If we could go to the next slide. The other features that we see in XDP are features of Parkinsonism, but there are some things that make it distinct from idiopathic Parkinson's disease. So what do we see from Parkinsonism? We see slow movement, small shuffling steps. People have a difficulty lifting their feet off the floor. Speech is affected. Handwriting is affected. And men may have both poor balance with a tendency to fall backwards and reduced arm swing. The 3 features that we see most consistently in people who have X-linked Dystonia Parkinsonism is the slow movement. A tremor can develop later in the course of the disease, and rigidity. So when we as the examiners move their arms or legs, we feel some stiffness or resistance to our movement. And we can go on to the next slide. So here are some examples of XDP. This is a gentleman with, by definition, generalized dystonia. Notice he cannot stand up straight, right? He's bent at the waist. He cannot straighten his arms fully. They're bent at the elbow. His fingers are also bent, right? He cannot straighten the fingers. And notice when he was walking, notice the big toe on the right is sticking straight up. He's walking on the outer edge of that right foot. So balance and walking becomes dangerous, right? So you can see only the left foot is fully flat on the floor. And the ankle and right foot are inverted and turned in. Okay. So this is one very typical example. We have blurred his face, but you may be able to tell that he has some uncontrolled head turning, right? His head tends to turn to his right. Okay. So this is a very classic, very typical picture. Here is another example. So here's a gentleman who has jaw opening dystonia and his tongue protrudes uncontrollably. The tongue sticks out. We are asking him to extend, straighten his legs. His eyes are closed. He has trouble keeping the lids open. He has to use his fingers to open his eyelids so that he can see. And the biggest issue in a gentleman with these symptoms is eating, right? Because it's very difficult to chew. It's very difficult to swallow. The tongue keeps sticking out, so the food cannot move from the tip of the tongue to the back of the mouth. And therefore, these men do become very malnourished. And I think we can go to the next slide. And here is another example. Here's a gentleman with significant dystonia of the neck. So you can see, right, that his neck does not remain straight. in a primary position. Notice his eyes are not blinking very much. That is a typical feature of Parkinsonism, right? This kind of stare. He has uncontrolled pulling back of his neck, and he's not closing his mouth completely. And so these are the best features that he shows. If we could go to the next slide, I believe there's one more. Here we go. Here we'd like to hear his speaking, I believe.
Oh, let's try that again.
So notice he needs help with several individuals to stand. And I think
Lovely yellow lilies. Newly laid linoleum.
Okay, so his eyes were staring open. He was not blinking very much. There was downturning of his lip. His mouth was slightly open. When he spoke, his speech was not distinct, and it also lost some of the normal variation in tone that we have in speaking. His voice was softer and more robotic and more monotone in nature. And he's a gentleman who cannot stand without assistance. So how do we think about who has Parkinson's versus who has X-linked dystonia Parkinsonism? Okay. One thing is that there's typically in Parkinson's disease, gait is affected and it progresses much more rapidly. In X-linked dystonia Parkinsonism, there's early bulbar dysfunction, meaning voice is affected more severely, more quickly, and there's significant difficulty with chewing and swallowing. Some gentlemen with X-linked dystonia Parkinsonism can have these significant inhalations. It sounds like they're gasping for air all the time, which we think is uncontrolled contraction of the diaphragm. And the other thing that is most, if you focus on the things, on the comments that are in red here, that we're finding are most consistent with X-linked dystonia Parkinsonism, is that the features of Parkinson's, the slowness of movement, the rigidity, it seems to be pretty much bilateral. We find it in both arms or both legs, whereas in plain old idiopathic Parkinson's disease, classically the features start on one side of the body. In idiopathic Parkinson's disease, the progression is much more slow, and we typically see not as severe speech and swallow impairment. Also, gentlemen with XDP tend to have more significant cervical dystonia, as well as dystonia in the hands or the feet that occur relatively early, within the first 10 years. Okay, we can go to the next slide. Okay.
Someone raised their hand. I don't know if you want to stop or.
Yeah.
Would you like to ask your question? Maybe that was unintended.
Okay. Okay. Well, feel free if anyone has questions, please. So based on our initial studies, what we are finding is that there seem to be different phases in X-linked dystonia Parkinsonism. So the most common phenotype is that first men develop dystonia, and it may be in their trunk, so it affects their gait, or it could be in their mouth and neck, or it could be in a foot. Then they hit a feature, then the Parkinson features become apparent. What is most apparent, though, when the Parkinson features first appear, is bradykinesia, slowness of movement. Okay? And the rest tremor is typically seen later. We then think for men who do not develop infections and do not become malnourished, and in fact are able to survive for a longer period, the dystonia becomes less apparent, and they display significant features of Parkinsonism. And what at that point, what separates them from idiopathic Parkinson's patients is the profound speech and swallow difficulty that they have. All right, we can go to the next slide. Okay, so what is there a difference in symptoms based on age at which a man presents? So at first the concern was, well, that perhaps we would see the dystonia earlier, but it turns out that that's not the case. But it's the Parkinson's features we see are the bradykinesia, the slowness of movement. I find it most useful to ask family members, you know, what have they noticed? Has someone's voice changed? Are they taking longer to get ready in the morning? Because typically the affected person does not notice that themselves. And then what we find is that there's a period early on, somewhere between 40 and 50, where the symptoms significantly progress, both the dystonia and the Parkinsonism, and then they settle out more. If we could go to the next slide. Okay. So when you see someone with some movement disorders, and there are, when do you start thinking about X-linked dystonia Parkinsonism? Well, if it's someone who knows that their family is from the island of Panay, certainly that should raise your suspicion. Although we have diagnosed individuals who, let's say, live in the greater Manila area and did not realize that several generations before their family had come from that island. Those who have a known family history of the disease, and this is an X-linked disease, and so it's important to review the family history and see if it's consistent, if there was any sign of a movement disorder or eating problem that followed an X-linked transmission pattern. We think, based on current data, that the age of onset is 39 to 40 years of age on average, and that there are features of dystonia or features of Parkinsonism or both. There is a question, is this disorder more common in the Philippines? As far as we know to date, all of the cases are in people of Filipino descent. It has not been found in other ethnic groups. And we can go to the next slide. Okay. So how do we diagnose individuals with XDP? Right? We obtain a family history. I usually just like to ask, you know, do you remember an elderly relative who had trouble walking or trouble speaking or eating? Because frequently in previous generations, this disorder was not, as with many other neurologic disorders, the disorder was not identified and properly diagnosed. Then a clinical evaluation by a neurologist, or preferably even a movement disorder specialist. And what we are looking for on exam are features of dystonia and Parkinson's. And then genetic testing is available to see if the affected individual has the genetic change that is associated with XDP. What are our current treatment options? As with idiopathic Parkinson's disease, all of the current treatment options are purely symptomatic, right? So the Parkinsonian symptoms can be alleviated mostly with using levodopa, dopaminergic agents, and also some anticholinergic agents. For people with significant jaw and tongue dystonia, zolpidem, also known as Ambien, can help, right? And that is also one of the most troubling symptoms, because people cannot talk and cannot have difficulty eating or drinking. For dystonia that affects a focal body part, let's say in the neck or a hand, we can use botulinum toxin injections. Keep in mind that the injections work for about 3 months and require then repeat injections. And then lastly, we can treat individuals with deep brain stimulation where there are different choices in the brain depending on symptoms, depending on disease, for where the deep brain stimulator can go. For X-linked Dystonia Parkinsonism, there's one specific region where we place the leads. The battery is under the skin, under the collarbone, and is adjusted in the office at like a regular neurology visit. So this is just to review. So symptom-specific treatment. So for dystonia, you can certainly reduce dystonia with anticholinergics. Trihexyphenidyl is the one we most commonly use, although I do believe that that is not available in the Philippines. There is another one known as benztropine that is available there. Benzodiazepines help with the dystonia. Most commonly, I use clonazepam because of its longer half-life. The limitation with these drugs always are the potential side effects. The sedation, anticholinergics can cause dry mouth. Alprazolam does not work as well, even though it's still a benzodiazepine. It is, there are differences in what receptors are activated by the different members of the benzodiazepine family. So it's really clonazepam or diazepam that are helpful. Another agent that can be used is tetrabenazine. Tetrabenazine depletes catecholamine in the presynaptic nerve terminal. So we think that what it is doing is reducing some of the firing in the brain that is telling the muscles to contract. Right? And then for particularly dystonia affecting the jaw and face, zolpidem can be of help. So botulinum toxin injections also provide some temporary relief. Typically, the effects last 2 to 3 months. And then for the features of Parkinsonism, we use the same drugs that are used in Parkinson's disease. Levodopa, carbidopa-levodopa is helpful. It can help to reduce the slowness of movement. And we use that one commonly. There are some newer drugs that are used quite a bit in individuals with idiopathic Parkinson's disease, and these are the dopamine agonists. We tend to stay away from them in people with X-linked Dystonia Parkinsonism because too much dopamine can actually worsen the dystonia features. So it's important to, and carbidopa, levodopa, or Sinemet is more gentle. And so it's important to adjust the medicine slowly so that we don't overshoot and cause other trouble. And then lastly, deep brain stimulation. But keep in mind that again, is just, these are all symptomatic. None of these treatments alter the course of the disease. All right, we can go to the next slide. What are some what are some complications of the disease? The biggest issue is swallowing. When people's speech is affected, typically their swallowing is also affected, and it is important to adjust diets, make sure people get proper nutrition, and certainly for the men who are here in the United States, peg tubes, feeding tubes are an option. Physical therapy and occupational therapy are also helpful, and it's helpful to send patients for PT and OT periodically throughout the course of their illness, because as their gait worsens, the assistance they need will change as well. And really, as with so many other chronic illnesses, nursing care is essential, right? It is the nurses who most are closest to the patients and most carefully observe changes in condition, including complications, pressure sores, or in the video you saw the gentleman whose big toe is sticking up, right? That can cause lots of trouble, can rub up against shoes, and cause pain and infections in the feet. All right. Now I'll turn it over.
I think this is Lori.
Okay.
So I'm going to talk about the genetics of XDP. So how is XDP inherited? So as Nuthan said, it's an X-linked disorder. So we have a little picture over here that shows, excuse me, that males have 1X and females have 2X chromosomes. And if the disease is on the X chromosome, then the male will express that trait and show the symptoms of the disease. So because the female has two X's, if she has the disease on one X and has a wild type or normal X chromosome, that wild type or normal X chromosome will mask the trait. And so she won't show the symptoms, or she may show symptoms later on in life, but she generally doesn't show symptoms, and she's called a carrier. And so because only females give their X chromosomes to their sons, you can never inherit the disease from your father. The males can't inherit the disease from their father. The sons can't inherit the disease from their father. They can only get the disease from their mother. And because mom has two X's, it's 50% of the time, each child has a 50% chance of inheriting the mutant X from the mother. So that includes and so when the son gets the mutant X, because they only have one, they express the trait. And when the daughter gets the mutant X from mom, because she, if dad is wild type, she'll have the wild type X from dad. And so she'll be also a carrier like her mother. But if there's an affected father, all of his daughters will get his affected X, and therefore they will all be carriers. Or if mom happens to also have the disease, then you could have an affected homozygous a daughter where she gets the disease on both of her chromosomes, and then she would express the trait just like her brother would. And so generally that's a rare occurrence for in a population, for a rare disease, to have an affected male marry a carrier female. But this shows us the timeline of the discovery of identifying that this was an X-linked disorder and getting towards the genetics of this. So as Dr. Sharma told us, in the 1970s, it was observed that there was a higher concentration of this movement disorder in the Philippines, specifically on the Panay Islands. And in the 1990s, several groups in Europe and the US, working in collaboration with their colleagues in the Philippines, collected families that had the disorder, and they did what was called linkage studies. So this was, of course, before the Human Genome Project, before we had sequence of the Human Genome Project. And so the way we would do linkage studies is we would take markers, variants across a region, and we would ask which variants segregate only in people who have the disease and not in people who don't have the disease. And by doing that, this gene, this disease, was linked to a gene in the beginning around the centromeric region of chromosome X. So both on the P and Q arms of chromosome X, so right around the centromere. And then as more families were collected, recombination events were identified that placed the gene into a smaller and smaller region. And so by 1999, the gene was placed into about a 350,000 base pair region on XQ1 3.1, which is what the green box is indicating on the X chromosome. So then in the 2000s, as next generation sequencing became available and genome sequencing became available, people started to sequence this region, and they identified 7 variants that were shared. So 5 of them were called DSCs, or disease-specific changes. There was also 48 base pair deletion. And then there was the insertion of an SVA. An SVA is a kind of retrotransposon. So these are elements that jump around in the genome, and one happened to jump into this chromosomal region. And so when you have markers that occur together on a chromosome, they're called a haplotype. And because we have in males only a single X chromosome, it's easy to actually visualize or see that haplotype, because you only have one allele at each or one variant at each of the positions. And so in the 2000s, it was identified that there was a particular haplotype that was found only on disease chromosomes. So the next slide shows that as part of the CCXDP Center, we used more of this next generation sequencing where we were able to actually capture. So we used a bunch of little primers that sit across, because we had now the genome sequence across this region, at least in Europeans, because of course the human genome sequencing was done in Europeans. And so we wanted to know what was on the Philippine diseased chromosome as opposed to what's in the European database. And so we could generate little primers going across this whole region. And so we covered a 463,000 base pair region of chromosome X, which is in the same place where we had these 7 disease-specific variants. And when we sequenced this region, we identified 54 variants in all that were specific to the XDP haplotype. And so over here you can see in the parentheses are the number of patients. So there was about 400 patients that were sequenced for this 463,000 bases. And you can see the vast majority of them had the entire 54 variants all the same, intact. So, but there were a couple of patients, and again, you can see by the numbers. So for this H2, there were 2 patients. And so these 2 patients have, from this blue, from where this red X is, all the way to the right, have the exact same variants as the people up here, but on this side, they don't have those variants. You can see they're green instead of blue. And so that is a recombination event that occurred that says that the disease, the shared region, is to the right of this X. And so we identified in, just a few individuals. You can see out of 400 individuals, it's like only less than 30 who had these different variations where there was recombination events in different places. And when you look at what is the region that is shared by everybody who has the disease, the region turns out to be just these 13 variants that cover about 200 kb. So we've come from 463,000 base pairs down to about 200,000 base pairs. And up here, this shows the different genes in the region at the top. And you can see now that in the red region, there's only this one gene called TAF1. And so this suggests that this gene must be the gene that's causing the disease. So I just want to reiterate, so when these markers, these variants occur together on a chromosome, that's called a haplotype. And this haplotype, these 54 variants, and now just this region that we're going to concentrate on, the 200 kb region, the 13 variants, they're unique to XDP patients. They're not found in any controls that we have looked at. So they're only found in patients who have XDP. And so what this represents is what we call a founder mutation. So there was one individual sometime back in time, and the mutation occurred on this individual's chromosome that had this haplotype, this background. And everybody who is descended, everybody who has the disease today is descended from that one individual. And so that is why this disease is much more prevalent in the Panay Islands of the Philippines, because that is where this person probably lived and had the first mutation. And so that is why this disease is pretty much specific to the Filipino population. The next slide. Go back for 1 second. So this last little thing popped up. So the other thing to know about this is that all the disease-specific variants are known non-coding. So in other words, they didn't change any amino acids. They didn't make a stop codon. They didn't do anything that is easy for us to say, oh, well, that could be the thing that's causing the disease. That could be the specific change that's important for the disease. So now go to the next slide. So here we just oriented, here's our X chromosome, here's our region of our haplotype. The red box is our disease-specific changes. At least the 7, not all 13 are on here. Then we have the gene, so you can see TAF1 is with this red box. And then you actually have the exon intron structure, so the coding, so the kind of little bars are the coding bits of this gene, and the arrows are the introns that get spliced out. And the little red triangle is what we call the SVA. So the SVA, it's made up of a sine, a VNTR, and ALU. That's where you get the SVA from. And it also happens to have a hexameric repeat at the end. And so this is just a piece of DNA that's repetitive DNA that, as I said, it jumps around in the genome. And so there's about 2,000 of these in our genomes, and they're jumping around. They're coming from I'm trying to find the word. They're coming from our ancestors. Like mice don't have these, so they're specific to apes and monkeys and what have you, and us. And basically, there was a Japanese group that had sequenced the SVA that was specific to the XDP chromosome. And when they sequenced it, they found this hexameric repeat had 18 different repeats on it. And we also cloned this region into a clone, and we sequenced it also. And we turned out to have 35 repeats in this region. And so that suggested that this repeat was actually polymorphic, that it varied. And so that was what we wanted to check next. Did this repeat actually vary? So the next slide shows that, in fact, this hexameric repeat, the length of this hexameric repeat is inversely correlated to the age of disease onset. So I told you that everybody has the exact same haplotype across this region, but it turns out that at this repeat within this SVA that's in this region, one of the disease-specific changes in this region, there is variation, and that this variation actually has a correlation, an inverse correlation with the age of onset of the disease, suggesting that this SVA insertion is actually somehow causing the disease. And this is also very reminiscent, if you know about Huntington's disease or the ataxias, the CAG, so-called CAG repeat disorders, where you also see this inverse relationship between the repeat size and the age of onset. And so in our case, the alleles range from 32 to 55 repeats. So although the haplotype looks the same, when you drill down into this specific region, you see that that these individuals actually have a difference, and that this difference, as I said, correlates with the disease. Next slide. So what does this all mean? XDP is a disease caused by a DNA repeat expansion, similar to Huntington's, Friedreich's ataxia, or C9orf72 ALS, and the spinocerebellar ataxias. The DNA repeat in XDP occurs in this type of junk DNA, so this repetitive region called an SVA retrotransposon, and this is abnormally inserted within the TAF1 gene. The XDP patients with longer repeats develop symptoms at an earlier age of onset, and the patients with shorter repeats may remain asymptomatic until later in life. And that this is what you can actually look for genetic testing. You can look for the SVA itself, you can look for the repeat, but you can do genetic testing now to identify who actually has XDP and has this disease-causing haplotype. And now Chris is going to take it over.
Just a few more slides. So what are we doing about it now? Well, we're doing a lot of things, more than we can really summarize tonight on this particular session. But one of the most exciting things we're doing is studying actually human tissue from patients with XDP. We founded the very first brain bank of any kind in the Philippines. And that was really made possible by our founder, co-founder Geraldine Acuna Sunshine, who literally went into the communities and said, this disease affects people I care about. Brain donation is absolutely critical to understanding this. That's a gift to research. Please join me in my mission. And by educating communities, we have built up quite an inventory. As shown here in this picture is Dr. Mark Ang, a neuropathologist based at UP Manila, PGH, and his staff. He came to Mass General and did a research training with us for a couple of months and went back. And our team based in Roas City fans out across the island and even the neighboring islands when they're alerted of donors who wish to pre-consent and give their brains to science. We've collected almost 100 brains actually at this point from primarily from patients and some from control relatives who want to give their brains to science also. That tissue comes to us at Mass General, and some of it goes to a brain bank in New Zealand. And we give that tissue away to people all over the world who study XDP. We have a very rigorous review process that vets those studies because we believe this is the most precious gift these families can give us, and we make sure that is used for the best possible experiments to help us understand the causes of XDP. And it's truly teaching us a lot. And this is a paper that we published with the Filipino team describing, this is one of the first brain banks in the world founded in a rural area like Panay, like Capiz, and the methods they had to put together to collect tissue. This is not, a morgue in an urban hospital. This is very different, and this is a consensus practice paper we published, which we've gotten tremendous feedback around the world from other brain banks trying to do similar initiatives. So we're quite proud of what the team was able to accomplish. This is a snapshot of our portfolio. I won't walk you through all of it, but basically we fund a lot of people do a lot of different kinds of things. We have people all over the world that are doing complex molecular biology. You heard Lori talk about some of it. I know the details get very wonkish, but this junk DNA that causes XDP is really complicated. And we have a lot of people who specialize in these kinds of DNA sequences that are asking, how does it screw up cells? How does it result in neurons in the brain dying? We have a lot of people doing different kind of cell biology studies, creating cellular models, which is actually what my lab does. How do we model it in a dish? We have people asking, how do we model it in animals? People making mice who don't normally have this kind of DNA sequence. What happens if you engineer it into them? People studying it in tadpoles and flies. A lot of interesting work you can do in these lower organisms. As I mentioned, a lot of people studying our tissue. And then Newton's team and other clinical groups around the world at doing clinical studies, neuroimaging, other kinds of clinical studies, natural history studies, most of which in partnership with major medical centers in Manila. And then interestingly, last year, we did our first hackathon. To be honest, I didn't really know what a hackathon was when this idea was suggested to us, but it's a competition. We invited people all over the world, primarily students, to give us their best ideas for how you could improve the lives of patients with XDP. We had 300 and some participants of over 30 countries, the most of which, number one country was India, and the second was the Philippines. Our first prize winner was a team from India who developed a solution for coding videos of videotaped neurologic exams. The second place winner were grad students from Manila, and they found something in a data set. We provided some of our research data, and they found a pattern that we hadn't actually seen, which is giving us some clues actually of a potential therapeutic strategy. And the third place team was also from the Philippines. They were actually from Cebu, I think it was, and they developed an app that would connect patients to their providers. It was a really exciting time. It was a great experience, and we hope to do it again. How can you help? Now I'm going to turn it back to Nutan.
So there are two ways in which we would really benefit from your help. One is thinking about, as you see patients, if you see gentlemen from the Philippines with features of dystonia and Parkinson's, most importantly, to think about this as a disorder, help them, get evaluated and diagnosed with a neurology team. We know that, many of these people live on different areas of the country. We are happy to help guide them to a local movement disorder center where they can get highly specialized care. That is our number one concern. And then secondly, if they are interested and willing, we would very much are trying to identify anyone with XDP and any willing family members, whether they have symptoms or not, and have them join our research study. And so those are the two areas in which we are looking for help from you, and also hopefully can be of help to you in getting patients the care that they need. All right. And so this is just information about referring people for care. And also, if you know someone who has XDP who's interested, this is our email address, xdp@mgb, which stands for massgeneralbrigham.org, and the phone number for our XDP research program, which you can certainly, and both the email and phone you can give to anyone who is interested.
There's also this.
Yes, and then this is, we have online advertising, and you can contact us through this as well.
And I think we are now done. So thank you. I haven't been following the chat, but it looks like there are a lot of comments. We are happy to take questions.
Great. Thank you, Chris, Newton, and Lori, for that excellent presentation. We don't have a lot of time for questions.
Yeah.
So let's just read a couple of the questions here. You were already able to address the first couple of questions. From Marilu, how can you describe the mental status of the affected individual, maybe distinguishing between early and late progression?
So we have not yet done systematic studies. There do seem to be some features more consistent with what you see in people with Parkinson's disease, in that they tend to become kind of more impulsive and have trouble with executive function, you know, keeping track of multiple things at a time.
Right. Thank you. I just saw a note here. Move the slide back for the email information, or maybe one of you could type in your contact information.
Yep. Yeah.
Or we can send the slides too, if that's okay with you. Just even the slide for the contact information and for the invitation for the clinical trials.
Oh, okay, great.
That you have.
And
Let's see. Any other treatments? And then I'll ask my own question related to that. So it does seem like a lot of what's going on right now is you're looking at disease biology. And from the hackathon, you did have one group with a potential therapeutic target. Any other options for like therapeutic targets or anything in the horizon?
It's too early for us to really disclose, but absolutely. We do have multiple candidates in development. Some of those are things that have never been in humans before, so that would have to go into safety trials. And some of the ones coming out of some of our funded studies are known drugs, and so their translation could be simpler. But that is our mission, and we are barreling forward. And it's part of why we want to partner with organizations like PNAA, because we can see clinical trials on the horizon, and that will require volunteers. We want everyone to know about it that may be affected by this disease. That we're trying to bring everybody together to test therapies to find something that works better than what we currently have.
That sounds great. I could see one hand up, but before I call Lanny, I think this is related to the question that we just asked. Success rate for the treatment of XDP, and there is no treatment that I have heard so far, and then mostly it's symptomatic, right?
Right.
And they're also asking about prognosis.
Right. And that it varies significantly. You know, I think that we don't have enough data to predict from person to person.
Yeah. Thank you. Lanny, did you want to ask your question? I see your hand up.
Yes.
Yep.
So I was diagnosed with cervical dystonia 5 years ago. And now it's getting worse. So if you have a cervical dystonia, will it progress? Or I'm getting Botox every 3 months, but it doesn't seem to help lately, especially after I had COVID. I had COVID a year ago. It seems like everything just got worse. So I'm just wondering, is there any other treatment I could use?
So, so a couple things. So one is that cervical dystonia is much more common in women. 70% of the cases are in women.
Oh,
And
okay.
it can, it does not, when it starts in adulthood, it does not typically spread, or if it stays in the neck and above, right? Sometimes people's jaws get affected. There are some oral medicines you can take that may help, although sedation limits you. And then, the other question is, has the nature of your dystonia changed? Does the dosing need to be looked at again, and the muscle targeting be looked at again? So those are the things to consider.
Okay. Oh, thank you so much, doctor. Thank you for all you do. Thank you.
So for the team, I have 2 questions here related to treatment. One is on gene therapy, the other is on deep brain stimulation, and I'll let you talk through that.
Want to talk about gene therapy first, Chris?
Sure. So gene therapy has become increasingly considered for a lot of genetic diseases. It has been considered for the brain. The brain is a little harder than other organs. Gene therapy for liver disease is a much easier target. You have to get a virus carrying something that you want to the right cells, and the brain is harder to transduce. There's lots of people work on it. In this case, a gene therapy approach would be actually probably gene editing. So you would be delivering like CRISPR machinery to cut out the bad sequence of DNA, and you'd have to get a virus to the right nerve cells and get the gene editing to work in a neuron. And that's a lot of technical challenges. We're actually sponsoring a conference in Boston in October of all industry experts who are working on gene therapy, CRISPR for any disease, because we're actually very interested to hear the state of the field, and is this something we should be investing in? We're investing some resources in it, but it's a relatively small part of our portfolio because we're not sure that it's really a viable option for this kind of disease yet. But we believe in leaving no stone unturned. So pretty much every possibility you could think of, we have on our radar in some sense.
Right. Maybe a quick word on the deep brain stimulation.
And then when do we do DBS? When someone is their walking is really affected, either, their trunk has a lot of dystonia, or their foot is really turned in and they are a significant fall risk. And the DBS tends to help quite a bit with the dystonia. Unfortunately, it does not help with the speech and swallow difficulties.
Great. It's I have a 6:01 here in my time, and I want to be respectful of your time. Thank you. Really would like to thank the team for doing the research on CCXDP. None of you look like you're Filipinos, but you're doing work for our countrymen. So we really would like to thank you. And we'll do our best to help advance the science and help you with moving the science forward. I was told that Dr. Chris Bragg will do the closing remark.
I will keep this brief because we're at the hour. You're right, we're not Filipino, but I speaking for myself at least, this has been truly a privilege and an honor for the past 10 years, and I think my entire team feels that way. And our partners that we've forged in the Philippines, who are on this journey with us, have also become, for me at least, some of my dearest friends. And I truly think that the work that we've been empowered to do by our co-founder, Geraldine Acuna Sunshine, who unfortunately couldn't join us tonight because she's in the Philippines right now, actually, is just some of the most memorable work that I think we will probably do in our careers and truly creating impact in the world. So I really hope that we've shared some of that with you. I think no one is more inspiring than Geraldine when she speaks about this herself. But we do our best to be her surrogates in her absence. And I hope that we've shared some of our commitment with you. And you can join us and help us take this word back to your communities and help us find people who need to be connected to the resources that we offer. You have our email address. Please reach out to us. We have more sessions coming up. And I'll be speaking in Asheville at the regional meeting in person for any of you that happen to be there. So thank you again for your attention tonight. We really enjoyed being here.
Great. Thank you. Thank you again. And, really looking forward to the second webinar in the series.
Yeah.
Mary Joyce, back to you.
Yep. Thank you everyone for attending. And just like what Marlon said, I'm sorry, I have a visitor interested in XDP. Just like what our president-elect mentioned, we will have 2 more webinar series for this. And Dr. Chris Bragg will close it during the Eastern Region Conference in Asheville this November. So thank you everyone, and we appreciate you. We will post all of the
Take a picture,
oh,
please.
picture.
Yes, this is our, if you will, beginning, the initial one.